I want to thank everybody for joining the MIDAS webinar for April. Today we are pleased to have Josh Colston presenting. Josh, I don't think I got a formal biography from Josh, so I'm gonna just sort of ad-lib off of his webpage here, where Josh is an epidemiologist, spatial demographer, and research assistant professor at the University of Virginia School of Medicine's Divisions of Infectious Disease and International Health, where he also teaches epidemiology in the Department of Public Health Sciences. Josh was trained with a PhD from the Hopkins Bloomberg School of Public Health, has a master's degree from the London School of Hygiene and Tropical Medicine. He spent several years working as a health analyst with international development agencies in DC and extensive work in Latin America. Has worked on, was a fellowship on the NSF Water, Climate, and Health Traineeship, and now he's working on a NASA-funded project to characterize environmental determinants of enteric disease, which I had no idea that was even a thing that NASA funded. Josh is going to talk to us about the Planetary Child Health and Enterics Observatory, which has me intrigued already just by the title. So thanks, Josh, for joining in. Really glad to have you here, and I'm going to hand it over to you. Please take it away.
Well, thanks very much, Harry. Yeah, thanks for the invitation to talk today. My name is Josh Colston. I'm at the UVA School of Medicine, so joining you today from Charlottesville, Virginia. I think that maybe that bio on my website needs updating a little bit. The NASA funding has come to an end, but we were very grateful for their support in that was the initial phase of this project, and we've now got follow-on funding from National Institute of Allergies and Infectious Diseases for this project, on which I'm principal investigator. That's right. It's called Planetary Child Health and Enterics Observatory. Enterics being the increasingly used shorthand for enteric pathogens, or pathogens that infect the gut and cause diarrheal disease. And so we're abbreviating it to Planeo, so pronounced like the Spanish word, which kind of has a double meaning. It can mean like I plan, or it can also mean soaring or gliding. A bit like a satellite, and we use a lot of Earth observation or satellite-derived data in this. So that's kind of the sort of levels of meaning to the name of this initiative. So yeah, I'm going to be introducing this today, and it's not so much a modeling analysis as a bioinformatic resource. So I'm not really a mathematical or computational modeler. Not yet anyway, although I kind of aspire to be one someday. I'm more of a traditional epidemiologist, so go easy on me with the questions, but I think this will be of interest to the group, because it is kind of a unique resource. Okay, so just a bit of background to diarrheal disease, which is kind of the broad area of infectious diseases that I work in. So if you were to take the causes of childhood mortality, so deaths in children under the age of 5 each year, and you were to order them, it would look like this. So sadly, still many causes of childhood deaths. You've got preterm birth complications causing the most, and then you can see here that at number 4 is diarrheal disease, the 4th leading cause of deaths in under 5s. Still to this day causing around 500,000 deaths per year. There's been a lot of improvements in that number over years, so that's come down a lot. A lot of that has been due to the rotavirus vaccine, which has been extremely successful in countries where it's been rolled out. But the rate of decline is decreasing, and that it's a stubborn cause of death, and it's remaining quite a contributor to that disease burden. And of course, with climate change, it's one of the main ways in which climate change is impacting human health, because diarrheal diseases are climate sensitive. But if you were to take that figure and pull it apart, so we're moving now from mortality to morbidity, or attributable incidence of diarrhea. And if you were to break it down by the etiological agent, or the bug basically that's causing the disease, the picture looks something like this. So I've kind of color coded the sections of this bar into the pathogen taxa. So you've got, in the shades of blue are viruses, the shades of brown are bacteria, and the shades of green are parasites. So it's quite a complicated picture. A lot of different bugs causing this syndrome, which outwardly kind of looks the same. It's not a differential diagnostic category, diarrheal disease. There are certain subtleties that clinicians can pick up on, telling you whether it's a bacterial cause or a viral cause, but not in every case. So as you might imagine, each of these pathogens has its own biology, its own sensitivities to the environment, and its own interactions with climate, host factors, household level, risk factors, that sort of thing. And these are quite difficult relationships to tease out in statistical models or in epidemiological studies. But nevertheless, that's kind of what we're attempting to do. So a bit further background, I believe that's an artist rendering of the Shigella bacteria there. Enteric infectious diseases, like I said, about 500,000 deaths in children per year, and that's mostly in low and middle income countries. Dozens of different environmentally sensitive organisms, viruses, bacteria, and parasites. And then the planet, well, so that's the Enterics and child health part of the name of our initiative. The planetary health part is, you all might be aware of this planetary health movement or transdisciplinary approach. So it emphasizes that, human health at a population level and even an individual level is bound up with the health of the planet and the Earth's natural systems, things like climate and biodiversity and deforestation and all these things that are moving in alarming directions. And so we sort of are applying that lens to diarrheal disease and looking at these planetary processes and how those influence the disease burden. And we take a big data approach. So, there's a lot of initiatives that we sort of compare ourselves to, or we aspire to be in league with, though we're not there yet. Malaria Atlas Project, the Johns Hopkins COVID-19 dashboard. Some of the people behind that are involved in our project as well. IHME has their local burden of disease as well as their global burden of disease project. Demographic and health surveys have their spatial data repository. So these are online repositories of spatial data products. And that's kind of what we're generating. So there's the component of fitting the models and making the predictions, and then there's a big dissemination component of making those results available in the same way that these more established initiatives have been doing for some time. World Pop is another one that you might be aware of, source of demographic data. So on the right of the slide here, you have a conceptual diagram that we've sort of come up with of generically how physical processes affect enteric infectious disease risk. This ignores the more proximal causes. Host factors, genetics play a role, feeding practices and nutritional status. So, setting those aside, though we do deal with them later, looking at the hydrometeorological processes, basically. So you've got atmospheric conditions. These have come into play a lot with COVID, since it's airborne and all these things affect like the size of the droplets that are always coming out as we speak and breathe, and how they disperse. But many enteric pathogens are also carried on airborne particles, and the same parameters can affect their dispersal. And then you've got the hydrological variables, soil moisture, surface runoff, so how the precipitation flows across the land following rainfall events. That can concentrate, lack of precipitation can concentrate pathogens in water sources. Extreme rainfall events can disperse pathogens in water. And then these sort of two levels interact in survival of pathogens on soil and surfaces, which can also determine infection risk. So the objectives of Planeo are to provide an evidence base for the targeting of enteric infectious disease specific child health interventions. So these might be vaccines. Some of these, well, we have a rotavirus vaccine already, but some of these other pathogens are targets for vaccines that are in various stages, but none, I don't think any are in phase 3 trials yet, though I may be wrong about that, but none are all that close to licensure. But there's other interventions like livestock management practices that are specific to pathogens, and interventions that generically target diarrheal disease as a kind of catch-all. We take a big data approach to modeling infectious disease, enteric infectious diseases. So we're compiling data from PCR diagnostics. So it's an acronym that if you weren't familiar with before the pandemic, you probably are now. That means, detecting the actual DNA or RNA of the pathogen in a biological sample. We use Earth observation derived environmental climate data, or Earth observation informed. So these models that take satellite data and as inputs to climate models, and these are often calibrated to ground-based measurements to give kind of a complete coverage prediction of different weather variables for quite spatio-temporally disaggregated, so you can get daily or even 3-hourly resolution. And we also taking data from household surveys. That's where we get the more household and subject level risk factor data from. If you're familiar with the demographic and health surveys and the multiple indicator cluster surveys, that's what those are. And we use geostatistical analyses with this. And then the goal is to establish a web-based dashboard and an international consortium of investigators. And we're sort of some of the way there with the consortium, less so with the dashboard. But it's a 5-year project that we've got 5 years of funding to
Josh, I want to jump in with a question from the audience. Who are asking, how does food as a transmission pathway fit into the conceptual model?
Food, yeah. So many of these pathogens are foodborne, like Campylobacter and things. Well, yeah. It's difficult to get data at the kind of scope and level that we're talking about on food exposure at the moment. But it's something that we're open to, and I know that the WHO is doing some work in compiling data on that. So it's an avenue for future exploration. Yeah, but you'll see the kind of data sets that we're working with, and how at the moment food exposure is a little hard to characterize. But excellent question, and if that was in the chat, I'm sorry, I'm not monitoring the chat. So do chime in if more of those come up. Good, yeah. So the epidemiological data that we're using is so there's this approach called independent participant data meta-analysis, or individual participant data meta-analysis. That might be an error there. And that basically means that, like, while meta-analyses use aggregated data and take them from multiple studies, in an IPDMA, you actually approach the study investigators and you ask for the microdata. So each row of the data that you get, rather than being an aggregate stratum, is an individual, or in our case, a stool sample. So samples of poop. These are studies that have taken stool samples and analyzed them for the presence of pathogens. So they have to have used PCR, because these diagnostics have different sensitivities. So we restricted to PCR, which is kind of the gold standard, to diagnose enteric infectious disease pathogens in stool samples. Studies that are carried out in low and middle income countries. So that's my area of expertise, and the data sets that we use are kind of only relevant, not so relevant to high income countries, though we're open to extending these approaches. We're primarily interested in preschool age children, so children under 5 years of age. These are the population that's most affected by these diseases. It's the population that they largely spend their time around the house and the peri-domestic area, so they're more exposed to the kind of risk factors that operate at that level. And so we identify these through literature review, kind of non-systematic at the moment literature review, and professional networks. We're lucky enough to be connected with a lot of investigators in different countries who are enthusiastic about this and willing to share their data. So we contact them with data access requests, and we have to go through a lot of paperwork, get all the agreements set up, assure them that we're storing the data correctly, since this is human subject data and everything. So there's a lot of admin that goes into this. So we get these study specific data sets, we combine them into a central database, and we try as much as possible to standardize the format and get all the variables consistent. And so far, I've just been processing a couple of studies that we've just been sent. So now we're up to 26 studies in 25 countries. So the map there is, I haven't had a chance to update it yet. But this was from a study that we've had published already. This was the bacterium Shigella. And so each of the circles there, the size corresponds to the sample size. You can see the locations of the study sites, and the area of the pie tells you the proportion of that were positive for Shigella. And then you can see a bit of the variety of study designs that we've got, which are what the colors correspond to. So, decent scope that we're getting here. Broad, range of latitudes, all, 3 countries where most of the low and middle income countries are, continents, I should say, where low and middle income countries are. And these samples are georeferenced, either to the households, but more often to, households is a bit of a headache when it comes to confidentiality and ethics and everything. So we tend to just georeference them these days to the village or the neighborhood where they live. So what you're seeing in this map is kind of aggregate to study sites, but within each site, we have coordinates that are dispersed over a little bit of a wider area. So people from different villages, or sometimes different households. And then, if those aren't available, if it was a health facility based study, we might just take the location of the health facility and say, that's as close as we're going to get. This is a proxy for the true residence location of the subject. Okay, so, we have this data process and flow kind of figured out. I guess I won't go into too much detail, but we get the data transferred to us. We store it securely, as I was saying. This is an illustrative map of how we might georeference households. Obviously, these are not real locations of real households included in this. It's just to kind of show you what we mean. And if you can imagine that we overlay, I'm sure everyone's familiar with, zonal statistics in GIS. You can get a raster of any that shows the distribution of any variable over the globe, and then you can extract the values to points. and we have daily, we're aiming to get daily predictions of enteric infectious disease risk. And so far, the approach we've been using is a Bayesian generalized multivariable modeling process. Though we're hoping to try different approaches. So to show you the kind of covariate data that we have, this is so we have a set of household and subject data variables. And the one I'm showing you here is one that, as a sort of side project that feeds into this, we're also modeling the risk factors of these enteric diseases. So we're taking survey data, and in this case, it's on finished floors. So that means the proportion of households that have something covering the floor of their house. So it's not just bare earth. They've got like wood or tiles or something. That's a risk factor because at bare earth, pathogens can propagate more in that sort of material. And of course, young kids are the ones who are scrabbling around on the floor all day, crawling around and playing with their toys there. So you can see how that would be a driver of infection. So we've got this model. We've modeled the coverage of finished floors across the lower middle income countries, and we're doing the same with water and sanitation variables. What else have we got? We've even got like livestock ownership, because some of these pathogens are zoonotic. They're can be transmitted from animals. So we've got like poultry ownership, the proportion of households that have chickens running around in their backyard, that sort of thing. So that's kind of at the most proximal space on the conceptual framework. Then we've got these static environmental variables. So basically, these are ones where we don't have it at a varying temporal resolution. It's just a snapshot. And the example I've chosen here is vegetation index. So just kind of tells you how lush the greenery is in a location. But people have used this in predicting all-cause diarrheal disease. So we think we'll be able to tell which particular pathogens are sensitive to that variable. Then the third kind are the hydrometeorological, so weather basically, but also soil moisture, which kind of isn't weather, but it's in the same kind of category. And so these are time varying. So what I'm showing here is an annual average soil moisture, but you can actually match so we know the date on which a stool sample was taken, and we know that we've georeferenced it, so we know the location. So we can match by location and date to the weather conditions that were at large when that sample was taken, and then we lag it a little bit because obviously there's an incubation period. So we aggregate over a 7-day window, and then we lag it by 3 days because these pathogens often have quite short incubation periods. And so we've published an initial analysis, like I said, of Shigella, and this was in the Lancet Global Health earlier this year. My colleague Hamada Badr at Johns Hopkins University, well, until recently there, he did most of the model fitting because he's written these R packages that make Bayesian modeling very efficient. So he was lead author on this, but you can see the number of authors that we have there. I think it came to like 50 something, because we like to give credit where it's due, and if an investigator shares their data with us, we try to include 2 investigators from each study site as a co-author, particularly if they are people who are from the country or from an endemic country, and that's one of the incentives, I guess, for sharing data with us, is that we include people as co-authors and give everyone credit for all the hard work that goes into actually collecting this data. And so we were able to, from this model, we were able to produce rasters of the predicted Shigella prevalence values. This is about a 25-kilometer resolution, so you all might be used to a bit more, higher resolution than that. It's a little bit coarse compared to some other products, but GLDAS, which is the climate model that we get our climate data from, is at that resolution, and we haven't been able to downscale it yet. But you start to see these hotspots a little bit. This is prevalence of Shigella in asymptomatic kids who are 12 to 23 months, so in the second year of their life. And we've identified these hotspots. As you'd imagine, sub-Saharan Africa has kind of sort of belt of high risk there. You can see little darker patches. There's one in Cambodia there, Papua New Guinea, Caribbean, Nicaragua. We're hoping to validate this, but the idea is that if someone is planning a vaccine trial or something, and they want a bit of a situational analysis of a particular location, or they want to decide on a location for a trial, they might be able to, this could inform that decision. Or if there was some other pathogen, Shigella specific intervention came through, you'd be able to target it to the most at-risk population. And so this is also an annual average. We were able to get sort of daily predictions based on the daily weather values for 2018. So you can, 365 of these. And we kind of aggregated it a little bit differently here to show the seasonality of Shigella. So panel A there is just the month of maximum probability. So you can think of that as the month in which the annual peak prevalence occurs. And you can see a nice, broadly speaking, there's a nice kind of hemisphere effect there, with southern hemisphere experiencing its peak around the turn of the year, and then more kind of mid-year peaks in the northern hemisphere. And then the panel B there is the amplitude of the seasonality, and that's the maximum value in a year minus the mean value to tell you how sort of pronounced the seasonality is. And that's kind of has some, some interesting stuff there. Like you wouldn't expect the northeast of China and North Korea there to be coming up as quite high amplitude, but it seems to. And then we picked 6 locations, just illustrative, a high and a low incidence location in each of the 3 continents. And you can see like the time series of the predicted values over 2018. These are so daily values, and then we've applied a smoothing method there to see the broad pattern. So if someone was interested in doing a Shigella study in Bangui, Central African Republic, they could extract those values in principle from that location and get a picture of what the annual pattern of Shigella is in that location. But as well as these predictions, there's this causal inference component as well. We want to actually most of these variables haven't been well characterized in how they affect Shigella. So we've got the prediction side, but also the causal inference side. Like what are the effects? What are the magnitudes of these effects? And we can actually rank the variables, as we've done here, with respect to how much they influence Shigella. And there's some quite surprising results. So improved sanitation facility is about 20% reduction in the odds of Shigella infection if you have an improved sanitation facility. Fine, that's what we might expect. But also practicing open defecation. So these are people who have no sanitation facility. They go in the field. Is also protective, almost as much. So what I take away from that is that having having no sanitation facility, no toilet, is better than having an inadequate toilet. But of course, the best thing is to have an improved sanitation facility. But that is kind of an actionable finding that could have implications for policy. Improved floor material is very very protective. And on the left hand side, we specified an interaction between soil moisture and precipitation, so you can see the shape of that in that sort of surface plot there. And then you've got the other time varying variables are in panel B there, and temperature was the most influential, and you've got it the predictions there plotted for different symptom status. Okay, so that's all the analysis component, and then we've got the dissemination component, which is really central to this initiative. Like I said, we're aiming to establish a web-based dashboard. So you'd have this map-based interface, data access and visualization system, suite of interactive maps to collate and disseminate the data products, and then we've got this international consortium of investigators that we're trying to foster. So that would be all the investigators who contributed data. We're going to try and have some kind of platform for all meeting, maybe like a webinar once a year, twice a year, in which we update them on the results and kind of foster some collaboration. So this is kind of how I'm envisioning the dashboard looking. So if you imagine this is kind of like a landing page, you've got your map-based interface here. So you can choose, you're going to be able to choose between, do I want to look at the observed pathogen prevalence, or do I want to look at the predicted pathogen prevalence? Let's say if you want the predicted, you can get these raster maps that I've been talking about. But let's say you're a bit old school and you don't quite trust those, and you want to see like what have people actually observed. Well, in panel A here, you can navigate to these blobs, and you'll be able to click on one and you'll be able to get a little summary of. So this is an actual study that took place. This was the SHINE study. It was a trial of water and sanitation in Zimbabwe, and it tells you there they found a Shigella positivity rate of just under 4%, and you've got a link to the citation there. So as another side project that's feeding into this, our division is doing a systematic review for the WHO, in which we're going to actually systematically document all the published estimates for each of these pathogens, so that we can map them like this. So it's going to be a repository not just of our model outputs, but also of kind of just the whole sum of human knowledge. Not quite, but it's an aim. Just everything that's been published on each of these pathogens together in one place for the first time. Okay. That's everything. This is the team. So we have my colleagues here at UVA. We have our Johns Hopkins colleagues, a number of different funding sources. All this has been ethically approved by the IRBs in both institutions. And we do have what I presented today. We do have a protocol that's under review, and it's available as a preprint in Research Square. And then there are some links to various things, my GitHub, my website, my Twitter. So with that, I guess I'll turn it over to questions, and thanks very much for listening.
Thanks, Josh. Really interesting. And I want to say before we get into any questions that I love the idea of what you're doing. I think it's fascinating. I also want to encourage, if anybody's on the line and has anything which is clearly in sort of a relatively early stage here, which I think this is, even though you've got this great paper out, clearly you've got a lot of work ahead of you on this project. And I encourage folks to be brave like Josh and to present things, even if they're not the finished product where you've got the big grand paper. I mean, it's a nice paper you got out there already, obviously, but
Yeah.
clearly it's just a starting point and far from the end point. So I'm gonna start off with questions and just ask if you mentioned aspiring modeling. Can you talk about what you aspire to do along the infectious disease modeling side and any predictive models or forecasts and what you think you would want to do with this data? Because I can imagine tons of possibilities, but I just would love to hear you say what you think is the interesting place to start.
Right. Yeah. No, I mean, that was part of one of my motivations, I guess, for doing this presentation is to sort of showcase this as a data resource to people who might know a little bit more about the kind of methods that could be applied to it. So, like I said, we've been using these Bayesian framework kind of models, and that was my colleague Hamada who did that. And it's been nice because you get these outputs that I, as a epidemiologist, can interpret, odds ratios, probabilities, and things. But you can also make the prediction maps. But then, we've got our colleague Samat at the Biocomplexity Institute that we were just talking about is in the early stages of applying some like neural network analyses to the norovirus data. And he has experience of developing vulnerability indices. So he can go from these neural network results to some kind of index of vulnerability. So that's a kind of analysis that is not spatially explicit. It's not using the coordinates in any way, but it's able to do something slightly different with the data. I think this is a, we're compiling this in a way that's generic enough that a lot of different approaches can be applied. And we obviously, what I've talked about today is predicting current prevalence. But we want to do some kind of like scenario modeling as well. What happens if you tweak these parameters? What happens if, you can take something from a climate projection and say temperature increases by 2.5 degrees in the next 20 years. What's that going to mean for pathogen prevalence? Some of them are going to go up, but some of them are actually going to, you might go down. Some of these viruses prefer sort of colder conditions. So, I mean, the obvious next step, is projections. Not just climate projections, but what happens if we do get a vaccine for Campylobacter? What happens if it has a efficacy of 90% compared to an efficacy of 60%? That sort of thing. Can we game out those scenarios? Very much open to suggestions. If anyone on the call sees this and it just screams out some particular method to them.
Fantastic. Well, I'm not seeing other questions here, and so I'm going to keep asking because I've got the questions, and hopefully other folks will join in. But I love the idea of talking about scenarios, and I'll get to that in a minute. But I wanted to just talk about a detail here that came up in my mind. If you could go back to the slide where you talked about the seasonality in those 6 locations. I was just a little bit curious. Yeah, that one there. I was curious about this because, I admit I am not a geographer, so I can't really look at this and see terribly well. But this is not really adjusted for latitude, is it? And so my guess is you have a few things here that are northern hemisphere and a few things that are southern hemisphere, right? And so how does that play out when you start looking at the seasonality? Because wouldn't you want to correct for, say, the warmer season versus the colder season, not just the absolute month of the year? Or is that not an issue and I'm just being naive due to my lack of knowledge of geography?
Well, no, that is a good question. So, we have daily temperature as a predictor in our model. So that's, in our points that are in the southern hemisphere,
Mm-hmm.
like mid-latitude southern hemisphere, that's going to be high, when on the other side of the equator it's low, kind of thing. So
Right.
the months of the year themselves were not predictors in our model. All I've done there is taken the predictions
Mm-hmm.
and grouped regions by their month of peak Shigella prevalence.
Mm-hmm.
So we're kind of letting the climate variables do a bit of the work for us, rather than being saying, like adding latitude as a predictor. We're saying that, like, temperature,
Mm-hmm.
we'll have that latitudinal gradient, but it will also capture some other things like altitude and things. Is that?
Yeah, sort of. I guess I was just wondering, so for example, on your graph A, that sort of has me looking at it and saying that's okay. So the stuff that's bright red in the south might be comparable to the stuff that's fairly blue and green in the north. And I realize that's oversimplifying because I'm not getting into altitude and other factors that are going to impact it. But I guess the question is that I want to wonder about is, if you take some place where it's, say, like in a given month, maybe you average 8 degrees C. How does that differ if you're in the southern hemisphere versus the northern hemisphere, or does it? And what are those other impacts or those issues? Is that an interesting question, or am I just overreading it here to say separating out the impact of the weather in a more fine-grained manner?
Right. So you're saying, is there a latitudinal effects even after accounting for all these variables?
Maybe, yeah. Or to put it another way, I mean, maybe there's something similar about what's going on in Southern Africa to what's going on in northern China in some unexpected ways that might tell you something interesting.
Yeah. I mean, it's a possibility for sure. I mean, all panel A is kind of a little bit of sort of kind of like a smell test to lend credibility to the results, because we sort of know that for a lot of pathogens, the southern hemisphere experiences its peak at the opposite time of year to the northern hemisphere. So what this is telling us is that our model is kind of correctly picking up on that.
Right.
But yeah, you're right. A way to go with this, and it's not something that that I know much about, but my colleague who I've mentioned, Hamada, has developed these regionalization methods where you can group areas. You can take all these outputs from the model, and you can say, like, what areas are similar to each other when you aggregate all these risk factors together.
Yeah.
And that would be super interesting if we found something like, there are these two regions that are, in totally countries that have totally different cultures and everything, and are across the globe from each other, but with respect to their sort of risk profile, they are similar.
Yeah, exactly what I was thinking, right? And you could almost imagine applying some sort of an unsupervised machine learning there to cluster regions according to some risk profile, agnostic of where they actually are, and then seeing if that tells you anything interesting.
Yeah, that's an excellent idea, and yeah, that'd be super interesting.
Models like that can often be a wild goose chase, right? You might do it and find nothing, but
sure,
it strikes me as worth thinking about, because what you'll probably get then is you might have regions that are both similar and then different in some informative ways.
Yeah.
Okay, intriguing. Okay, I'm fascinated.
Yeah, excellent question.
Okay, so hearing the other questions, I've got two other biggies that I wanted to ask you about. So the one thing that I'm intrigued about is firstly just mechanics here in terms of the data. We've spent a lot of time thinking about data in the scenario in the MIDAS Coordination Center, and you said you've taken a lot of data that is sensitive and done a lot of work with data use agreements, and I feel your pain. That is a long slog where you end up feeling more like a bureaucrat than a researcher as you go through this. And so I commend you for your stamina and pushing through that. But are these data then, once you've processed them through, are they available in some form that others could get their hands on without going through some of that? Or do you have some plans for sharing the data with the community beyond just a dashboard?
So the input data, the training data, we're just not able to, unfortunately. I mean, it's 26 different studies. Some of these data have been made publicly available already. So the MALI-D, GEMS, and SHINE studies, they're already in a repository called ClinEpiDB. But some of these other studies, they were a little bit, I had to do a lot of convincing to get them to share just with me. So we're just really not able to get sort of the permissions from all of these studies to put it out there. So if someone wanted to get access to the raw data, we would have to set up an IRB, and they would have to be probably get them as a visiting affiliate of UVA or something like that.
But what about sort of your downstream results, some of the model outputs, for example, that might say do predictions of things over time, or some of those things might be useful for people to work with in a secondary sense.
Yeah, so we're gonna. We'll make the rasters available for download. In fact, you can already get these ones in my GitHub repository, and so you'll have the interactive part of the dashboard where you can click on a point and it'll tell you the prediction, but also you'll be able to download it as a raster file as well, the prediction. And people might use that as a further input to another model that they're working on.
Okay, great.
Yeah.
Okay, there is a question here in the Q&A. Thank you, Sean Moore. Given the difficulty in estimating the attributable fractions of different enterics to disease burden that you mentioned, how important do you think it might be to consider multiple pathogens simultaneously versus modeling them in isolation?
Yep, excellent point. These are pathogens that exhibit co-infection frequently. It's not uncommon for a child with diarrhea to come out positive for several pathogens at once. There's a lot of questions starting to be raised and a lot of research about how these interactions operate, and it's something that we want to explore and we're interested in. There's a little bit of a practical problem in that not every study in our database tested for every pathogen, so we don't have we're focused on 10 pathogens, but we don't always not all of our rows are complete when it comes to pathogens. So it's a little tricky, but using one pathogen as a predictor for another pathogen in amongst all the other predictors is something we want to explore. So thanks for that question.
Okay, great. And I'm going to ask you one more about the you mentioned scenarios, and just as we spend a lot of time with MIDAS with the scenario modeling hub, and so this is near and dear to my heart. Do you have any thoughts on what sort of scenarios you would be most interested in exploring?
I mean, we're interested in climate change scenarios. So what happens if you increase the variability in rainfall, something like that, something that's, plausible, we can plausibly see over the coming decades. What happens if you raise the temperature? What happens if things dry out a little bit in some areas, but get wetter in others? What would happen if, increase the coverage of finished floors by 10%? And then, like I said, the vaccine scenario, things would get a little bit more complicated when you start adding vaccines in. The relative contribution of other pathogens would go up, and the odds of it causing diarrhea would go down and things. So trying to tease out those sort of complicated effects of tweaking one parameter, how does that affect the other parameters, that kind of thing. But those are the main ones. Climate change, just general development, increasing coverage of sanitation and things, and vaccines and other interventions.
Interesting. Fascinating. But with the climate, it seems like you could both look at changes in absolute values, say temperatures increasing over time, and the variability are both factors that could play different sorts of roles, correct?
Exactly, yeah. So we'd have to, and then, so the obvious ones are temperature and rainfall, but then there's things like, soil moisture. How is climate change going to affect soil moisture? Well, it's going to affect it differently in different places. It's going to diverge. What happens then? And it's going to be different with different pathogens, because, like I mentioned, these viruses tend to prefer cooler conditions, and then the bacteria tend to prefer warmer conditions. So, how eventually we want to take into account like the whole pathogen profile of the location, so how is it going to be like the bacteria are going to displace the viruses, or the other way around, that sort of thing, so there's, the individual pathogen picture, and then when we've got all of them, we're going to have to look at, just the whole profile. Fascinating.
Well, I think this is a good point to wrap up. We don't have any questions on, and I've asked more than enough. But this is really nice work, Josh. I think it's fascinating. I love to see the effort in coordinating this much data from this many places and trying to make something of it. I might be getting in touch with you with a couple of extra questions, but for now, I want to say thank you so much, and thanks to everybody for coming. And hopefully we'll see you all in May at the next one.
That's great. Thanks a lot for the opportunity. Thank you, Harry, for all the organization, and thanks to everyone for tuning in.
Our pleasure. Take care.
Take care then. Bye.
Bye.